All right, well, welcome everyone. Thanks for joining our EV Journal Club today. This is the 7th session that we've had since the COVID-19 pandemic shutdown. And I'm delighted to introduce Mike Paulaitis today. He is my colleague at Johns Hopkins University, and we've been working together on several projects over the last few years. But before I introduce Mike, just some housekeeping notes here. Those of you who have attended the Journal Club before know that we like to use the chat function to make comments, but also to present any questions that we have. So we're keeping people muted until the end of the presentation, but you're welcome to introduce your questions on the chat feature. And then I and some of our lab members will try to compile those, and then we will unmute you towards the end so that you can interact with our speaker and ask questions. I also just wanted to note that that the International Society for Extracellular Vesicles meeting this year, our annual meeting, has been canceled. The in-person meeting, which was to have taken place in Philadelphia this month, and instead it has been rescheduled towards the end of July. There's going to be a 3-day virtual event, and this will be followed by a 60-day period where you can access all of the content. So there is a nominal fee for participation. The registration fee is pretty low for ISEB members, and you'll be able to access all of this content for about 2 months. So please visit the iseb.org website if you are interested in signing up for that. So now I would like to introduce Mike Paulaitis, who is our speaker today. So Mike is a professor of ophthalmology at Johns Hopkins University, and specifically in the Wilmer Institute, which is our institute that studies the eye. And Mike in the past was actually the head of chemical engineering at Johns Hopkins, and then he was at Ohio State University for quite some time. And so he has been working on microRNAs and on extracellular vesicles in recent years. And the paper that he's chosen today, we're very excited to hear about this, because it addresses some of the big pressing questions in the field right now about EVs and how we might be able to scale them, to load them. And so without further ado, I'd like to hand this over to Mike. So thanks, Mike, for joining us. Thanks for presenting, and we look forward to learning from you today.
Thanks, thanks, Ken. First of all, I smile every time somebody introduces me as a professor of ophthalmology. I'm a chemical engineer. I'm in the Center for Nanomedicine in the Wilmer Eye Institute, which is a center that is focused on drug delivery using nanoparticles. The faculty are mostly chemical engineers, but it's a highly disciplinary center, and that's really how I started interacting with Ken. Ken did mention, I'm going to go to full screen now so you can see my slides. So Ken did mention that I was at Ohio State for a while between my first tenure at Hopkins and now. And one of the reasons I chose this paper is because the senior author on this paper is Jim Lee, who at the time I was there had a nanotechnology center. I was part of that center, and we actually started our work in exosomes together. I worked with him on his tethered liposome array chips. This project of cellular nano-electroporation was a separate project at the time. In fact, though, when it was first introduced by Jim, it had quite a few shortcomings. One of them was that it wasn't a high throughput. He was moving cells around with optical tweezers, and it was mostly a single cell assay. So when I saw this paper, I realized that Jim had finally gotten over that obstacle. And the other thing that intrigued me about this paper, which I've written down there at the bottom, is that this technique, which I call cellular nano-electroporation, can generate large number of EVs with high mRNA loadings. Anybody in the exosome field should be their ears perk up every time they hear large numbers of EVs. It's not in our DNA right now to expect very large sample sizes. Then the other intriguing thing, when I started to read the paper that made me interested to learn more and present it, was this contention that these large number of EVs with high loading was generated through exosome biogenesis mechanisms that were actually impacted or caused by the cellular nanoelectroporation process itself. So we'll go into that. Let me start then with an outline of the presentation and the paper. So I'll give one or two slides of a brief introduction to cellular nanoelectroporation. I think it helps explain some of the data you'll see afterwards. It's in the paper. And then the next 3 bullets essentially follows the paper itself. We'll look at how they characterized EVs, the most important characterizations being the number of vesicles or EVs per cell that's released by these cells and their mRNA loading. The issue of whether the mRNA is loaded in the exosomes versus the microvesicles, the impact of this method on the mechanisms of exosome biogenesis, and then how they generated therapeutic mRNA-containing exosomes using this method, and finally their demonstration of the therapeutic efficacy of this technique. So how does nanoelectroporation of cells work? And what I've shown here, some of Jim Lee's earlier work, which if you look in the top left-hand corner is one of these single cell chips in which a cell is loaded on one side. And as I mentioned, he does this by moving it around with optical tweezers and making sure it's located right above the nanochannel in that cell, or I guess I say orifice. And the other side is a gene or drug is loaded in there, and then the two are connected by a nanochannel. The middle diagram shows you an actual visual picture of the cell loaded. The nanochannel then is shown on the right. In these earlier manifestations of this instrument, the nanochannels were about 95 nanometers in diameter and 10 or 15 microns long. What happens in the nanochannel then is there's a power supply that pulses a voltage across that nanochannel, and the gene or drug of interest, in the case today it's going to be DNA plasmids, will move across the nanochannel and be transfected into the cell when the cell membrane porates. So how does that work? So this bottom left-hand diagram gives you a basic idea of how the poration and then transfection works. And so this is the cell, the nanochannel, and the reservoir with the gene or the drug. And since we're putting a voltage across here, this is nothing more than an electrical circuit. And you can think then that a pulse of 200 volts for 10 microseconds across this channel will have a high resistance right at the nanochannel cell membrane, shown here with a capacitor. The voltage then, as it builds up to a certain threshold voltage, will cause the membrane to porate. Then the circuit's complete, and then the next resistor is the other side of the cell membrane that again it can be a resistance to the voltage and can actually porate over there. Although in this work we see that a little bit. I think that from what Jim tells me, that can be controlled. So this large voltage buildup here, poration, then moves charged species, in this case the DNA plasmid, across either by drift or a large electrophoretic drift. And it's this large electrophoretic drift that moves the DNA into the channel and into the cell. That's shown over here on this slide, where here's a cell before poration at time 0. The white line is the actual nanochannel. And then 30 microseconds after nanoporation, the pulse lasts about 10 microseconds. The cell essentially fills with, in this case, propydium iodide dye. And then over the next 2 seconds, that dye diffuses throughout the entire cell. In comparison, this is bulk electroporation. Again, the cell. In this case, you can see the time frame is much longer. It's 150 seconds, and it's more of a just strictly diffusive process, and probably endocytosis too, since some of the genes of interest aggregate on the surface. So this process is fast. There's almost a linear response between the pulse rate, pulse length, and dosage. So it's also a controlled transfection process. That's the old version. The new version does nothing more than eliminates that single cell pore, and now just puts the cell itself down on an entire array of nanopores. And then the cell will sit on one side. This is upside down. The cell sits on this side here. The reservoir is filled with the DNA plasmid. The voltage is applied in pulse, and we're off to the races in terms of transfection. So how does this work? So here's their first set of results. And this shows cellular nanoelectroporation in, for different cell lines, mouse embryonic fibroblasts, bone marrow dendritic cells, mesenchymal stem cells, and HEC293 cells. And in each case, they're looking at the transfection of 3 plasmids. They're roughly 6, 7, and 9 kilobytes in size, kilobase pairs in size, sorry. And they're comparing different transfection mechanisms. So control, PBS, lipofectamine, bulk, electroporation. Electroporation, where all 3 of the plasmids are in the solution that's transfected into the cell. And then electroporation, where they're just using PBS as a control. And that's in all 4 cases. And the clear thing you see from this is, in all cases, the electroporation is much, much higher in the number of EVs produced per cell. This is 10 to the 4th, so we're 100, we're talking about 1 million EVs per cell for the mouse embryonic fibroblast. This is about 5 billion. Here it looks like about 6 million if I can read the exponents, and here it's about 3 million. The other thing to notice is the electroporation itself, even without the plasmids, causes a large number of EVs to be produced by these cells. So we can also compare, or they also compared their nanoporation to other techniques for transfecting cells. In this case, again, the number of EVs per cell, and the cellular nanoelectroporation, again, is about at 100 or a million EVs per cell. And then starvation, hypoxia, and heat. Again, there's a break in the scale. So the starvation would come in is the highest, and it comes in around 75,000 EV numbers per cell. That is quite a high number from my experience. And I'll come back to these high numbers in a minute. But clearly, 1 million is, even if you disregard the high numbers relative to 75,000, is a big increase in EVs released by these cells. This shows the time course of emission. The peak emission is around 8 hours. From what it says in the method section, these EVs are collected over a period of 48 hours. We can also look at the loading in these cells. These are the those 3 plasmids I mentioned before that produce transcription factors. This actually came from older work that Jim was doing, where he was transfecting these cells with these 3 transcription factors to transform these cells into neurons. And I think what happened in that earlier work is he noticed there were a lot of exosomes being produced, and that led him to this study. So again, fold change in terms of the mRNA that are in the vesicles, the EVs, they come off of these cells, much higher for the nanoporation in each case than for bulk electroporation or lipofectamine. This shows the time course. The faster, the smaller mRNAs get taken up faster. And this shows that protein translation, that these mRNAs are functional. In this study, he's actually going to look at therapeutic effects of P10. So he shows the same thing now for the P10 plasma being taken up into the exosomes, and then these are the fold changes of the mRNA in the EVs themselves for both the mouse embryonic fibroblasts and the bone marrow dendritic cells, about a 2,000-fold or increase. Gel electrophoresis shows, again, confirms the P10 plasmid transfection gives the highest amount of P10 in the exosomes, whereas if you just put P10 in solution and transfect it, it's lower, and then this is the control with the poration without anything but PBS. So let's come back to this issue of these very large numbers of exosomes being produced. The statement was made after that first slide where he showed the numbers that the vesicle size distributions for all of those studies were the same. And then in the appendix or the supplementary material to this paper, he shows this plot of size distributions. The figure caption isn't that clear, but the way I interpret this is this was nanoporation of just PBS, and this was nanoporation of the 3 plasmids. Both peaks are bimodal, so we see a population of larger vesicles in both cases. The mode is around 260 nanometers in diameter. The smaller population, though, shows a shift. For the PBS group or control group, this peak is roughly around 35 nanometers, whereas for the nanoporation with the plasmids, this peak shifts to around 70 nanometers, where we would expect to find EVs. And I asked Jim via an email. He's stuck in Taiwan, by the way, at a because of COVID-19, he can't get back into the U.S. Why is there this shift? And his comment was he thinks the shift is due to the fact that these small vesicles have a lot of mRNA in them where these don't. And so larger vesicles are produced. It sounds like an opinion now that has some preliminary data. I asked him if he knows the density differences between these. He replied it was a good idea to check it, but that's his current opinion of why there's this shift. The other thing I want to come back to, though, is every measurement of number of vesicles per cell produced is a DLS measurement. And again, I asked Jim how we can get absolute numbers of particles released by exosomes or released by cells, either exosomes or vesicles from DLS. And he said what they do is they use nanosite and standard beads to calibrate their DLS measurements. He promised to send me the methodology for that. It's not in the paper, but I suspect that those very high numbers for starvation might be due to that calibration, might be buried in that calibration method itself. So I'll leave that there and then go on to the obvious question. If there are two populations, where is the mRNA that they're seeing in those previous assays they did? So they took, now by ultracentrifugation, the 10,000 K spin to get what is called the microvesicle fraction, and the 100 K spin, or 10 K spin and the 100 K spin to get the exosome fraction. And you can see peaks here of around 260 again, and this one looks like could be around 70. They looked for exosome markers in the exosome fraction, which they found, and a microvesicle marker in the microvesicle fraction and not in the exosome fraction. They did preliminary nanosite, which shows about a 4-fold higher level of total RNA in the exosome fraction versus the microvesicle fraction. They also had cryo-TEM images, which shows when they porate with their plasmids that produce mRNA, they see it in the vesicles in the cryo-TEM, whereas the vesicles with just PBS are empty. And then finally, they did an OptiPrep fractionation, and they see that most of the RNA is in the fractions that would correspond to 1.10 to 1.15 grams per ml densities, which is the exosome fraction. At this point, given the practical focus that Jim had in this paper, that was the last OptiPrep fractionation they did. At this point, they used ultracentrifugation to separate exosomes and microvesicles. So let's look at the mRNA a little more closely. Again, going back to these three transcription factors, the mRNA for them by qRT-PCR, again, much higher in the exosomes in each case than in the microvesicles, and the mRNA, I think I might be saying microRNA, I apologize for that. Everything's mRNA. The mRNA is actually functional in that it can be transcribed into proteins. And then this image is an image of Jim Lee's tethered liposome array. And what it shows is in this array, tethered to the surface, are liposomes which have 3 molecular beacons for these 3 mRNAs. And so when an exosome binds to the surface, fuses with these liposomes, the molecular beacons light up, and he knows, and you can count then the number of exosomes that have the RNA in them, the mRNA in them corresponding to that molecular beacon. There's a color code here. The pink arrows are arrows which show only one molecular beacon lighting up. The blue or turquoise arrows show where two molecular beacons are lighting up. In other words, those exosomes have in them two of the mRNA. And the yellow arrows show where all three mRNA are in the same exosome. And this was one of two images he shows. This is one where he actually porated each one sequentially to show that he can optimize or get a large number of exosomes with all three plasmids in it. That will become important later in his therapeutic studies, because he's going to add multiple plasmids to his exosomes, essentially to engineer them to both target certain cells and to transfect them with a certain mRNA. So that's basically the data. What's the mechanism then that he's applying here, or that he thinks applies here to these high numbers of EVs being produced, exosomes being produced by this applied voltage across this cell? And he proposes two mechanisms based on work in the literature. The first is when these pores form, not only do the plasmid, the DNA plasmid go into the cell, but some of the fluid goes into the cell too. So there's an uptake of calcium ions across that pore during poration, which then is known to stimulate exosome release. The second mechanism is that resistance, that high resistance at the cell nanochannel interface causes a temperature rise. And that temperature rise is a heat shock, which produces heat shock proteins, which in turn can upregulate p53, and p53 can upregulate TSAP6, which has also been shown by some elegant work by Arnold Levine that this pathway in these particular cells that he was looking at, the mouse embryonic fibroblasts, leads to higher exosome release. So now he goes about looking at both the effect of calcium ion uptake and heat shock and the formation of heat shock proteins by this pathway on his nano-electroporated cells. So the first thing he does is he shows that the MVBs increase with nano-electroporation. I'm not so convinced by this, and I'm sure it can, because this is a PKH26 dye, would have doubts of this as well. What he's showing is that with nano-electroporation, there's a lot more dye fluorescing, and therefore there's a lot more vesicles being formed. The implication is these are increased numbers of MVVs. Here he entered in the comparison in B, he's actually transfected using nano-electroporation the cells with GFP labeled CD63. And again, what he's saying is that we see more MVBs, an increased number of MVBs with this electroporation versus bulk electroporation because of the high concentration of the CD63 at different spots. I frankly don't find either one of these that convincing. I've seen people do the TEM images, and these results seem more convincing to me. And that is, let's just take TEM images, count the number of MVBs, and while we're at it, we can count the number of of ILVs in the MVBs. And doing that, he sees higher numbers of MVBs and larger numbers of ILVs in the MVBs with nano-electroporation. And he also sees several proteins related to exosome biogenesis being upregulated with nano-electroporation. So that this set of results seems to be almost to be a reviewer's comment, a response to a reviewer's comment, because then he goes on and does the obvious things. Is he does the practical approach, what I would have expected Jim to take, to look at what happens in these cells when he changes the amount of calcium ion in the buffer. So the top row here shows the duration at which these pores in the cells last. So at each time point, 0, 15, up to 300 seconds, propylium iodide is added to solution, and you can see a large uptake by the cell at 0 seconds, right when poration occurs, and then these pores eventually close after about 2 minutes. And the cells themselves stay structured with no with breakdown. And then the results are that when he increases the amount of calcium ion in the buffer solution during electroporation, the amount of intracellular calcium ion goes up, and again, the amount of exosomes produced, here's our 1 million exosomes standard, as we increase the calcium ion concentration in the buffer solution, and the effect is essentially linear. Number of exosomes produced per cell versus the intracellular ion concentration. He can block the calcium ion with a chelator, or the effect of the calcium ion with a chelator EGTA. And these are the same results, except now he's adding EGTA, and he sees a reduction in intracellular calcium ion with electroporation, and he sees a reduction in the number of exosomes. This to me is not quite as large as I would have expected based on the previous results in this reduction. And in fact, it might be an indication that there are other mechanisms that need to be considered here. Local heating, the second part of his mechanism, and in this case, he adds a rhodamine dye, a red dye, and you can see them in the channels. This is at time 0 before electroporation, and then with electroporation, the rise in temperature, they disappear. Top view, the same thing on the side view. So this is the nano channel at 0 seconds prior to poration, at poration, and then 1 second afterwards when the dye starts to return its red fluorescence. The estimate of the actual temperature at that interface is around 60 degrees. He also sees upregulation of heat shock proteins relative to the control, and when he electroporates with inhibitors for the heat shock proteins, he sees a decrease in the number of exosomes produced. He goes a little bit further to pursue this P53 TSAP6 signaling mechanism as being the process. He has mouse embryonic fibroblast cells, wild type and where P53 is knocked down. So again, he sees when he electroporates a large number of EVs produced per cell, whereas in the knockdown, he does not see that large increase. Again, Western blots show P53 protein corresponding to the high number of vesicles, and the TSAP6 protein as well. P53 upregulation, though, has no effect on apoptosis, as shown by these flow cytometry results for annexin 5 and cytochrome C and calnexin. So, well, at this point, let me go back to this slide and make one other point of emphasis here. He's saying that both of these together are what is producing his large number of EVs plus mRNA loading in the EVs. One or the other might produce more exosomes, but the loading too is a critical factor. And so it's both the calcium ion uptake and the heat shock that produces these high number of highly loaded vesicles. So let's go on to his therapeutic study. The first thing is to generate these therapeutic exosomes, and what he does is he adds a targeting peptide for either the GL261 glioma cells or the U87 glioma cells. He shows by a pull down that these targeting peptides are outside the exosomes and not inside the exosomes. He actually produces these exosomes, which he calls XOTs or tagged exosomes, by taking the plasmid for CD47 with the targeting peptide on it and electroporating the cells with the plasmid. In these series of fluorescent images, he's showing, again, uptake of the targeted exome with a CD47 targeting peptide being higher than the exosomes without the targeting peptide. And then he's also added at this point the plasmid for P10 and shows that more P10 is in the exosomes that are targeted for the, in this case, it's the glioma cells, the GL261 cells, than the exosomes themselves. And this is just quantification of that data. Takes that 2 steps further to look at the mechanism of uptake, and what the conclusion is by, again, looking at PKH26 dyed exosomes and various markers for clathrin-dependent endocytosis. This is caveolin-dependent exocytosis, and macropinocytosis shows that it's clathrin-dependent endocytosis is the predominant mechanism. And he also confirms that by showing greater inhibition with sucrose than the inhibitors for the other 2 methods. He shows the cells are viable. In this case, it's the GL261 glioma cells. In a mouse model, he shows better circulation longer circulation of these exosomes when they're targeted, either just with the peptide or, I'm sorry, I can't remember what the C is. This is the targeted peptide is more than the exosome itself. And then there are no toxicity or immunogenicity effects of the targeted exosome. So then on to the mouse model. So tail vein injection of mice with these exosomes. In this case, he's looking at these are the results for the U87 glioma model. Find the targeted peptides end up more in the brain than for the exo T versus the lipofectamine, the PBS, or the exosomes without the target on them. Again, the same results here for the targeted exosomes, and this is the quantitative results showing more targeting to the brain with the CD47 targeting peptide. And these results are to show that the targeted exosome really does get into the tumor tissue and is not just in the blood itself. The mice then show with the XOT much slower tumor growth than either the control, the exosome without the target, the empty target without the P10, and again, the turbofectamine. Again, that's shown here, and then the survival time of the targeted exosomes mice are extended. And then finally, this is I guess these are histographs of the tumor cells themselves. Shows upregulation of P10, downregulation of the signaling Ki67, which shows the tumor cells aren't proliferating, and then this is staining their nucleus and extracellular matrix, which shows that is also diminished, so that the P10 is getting into the tumor, and it does appear to have effect on tumor growth. He also looked at, so this was one set of results. There's a second set of results for a second mouse model. They're virtually the same, so I didn't really think it was worthwhile going into a second set of data. Let me then just conclude first of all, I found the major conclusion of the paper, and this is just the restatement of what I said on the first page, that this method generates large numbers of EVs with high mRNA loadings, and it does that by actually manipulating or impacting the exosome biogenesis mechanism through the electroporation process itself. And then the practical side, this is taken straight out of their discussion, is they've demonstrated this process as being a robust process for producing therapeutic exosomes from multiple cell sources. So first of all, given that the paper itself has a practical nature to it, there's still some pretty, and for that reason, there's a lot of basic research that could be done, particularly on the exosome biogenesis side and the EV side. And these are the first two questions I mentioned here. There's still big effects that they're seen. So are there other mechanisms that are possibly involved besides the one they looked at? And of course, are there other carriers that he or we would want to consider if Jim gave us one of these pieces of apparatuses to play with ourselves? But I think from a higher level, these are interesting points to also discuss, and that is, I showed how this electroporation technique compares to starvation, hypoxia, and heat shock. And is it then an alternative, potentially more versatile mode of inducing cellular stress under different controlled conditions that allow us to look at stress response in terms of EV or exosome production? And then the last thing is the discussion really talks about large-scale production of therapeutic EVs, but really when you talk about these 1 centimeter by 1 centimeter chips, that still isn't a large scale up. And it seems to me that this might be an interesting translational research tool rather than right now a tool for commercial production of therapeutic EVs. And then that question is, if you had to play, if you could play with this, how would you play with it? So let me stop there and open it up for Ken's questions.
That's great. Well, thanks so much, Mike, for the presentation. And we have had a few questions that have come in during the course of the presentation. So I'd like to start here with Brian Bird. So Brian, you had some questions about endogenous mRNAs, and I think that some other folks also had some questions. So Brian, I am going to let's see. Actually, Brian, I do not see you. Are you on here under your own name? All right, I'm going to actually then ask the question that Brian he is under his.
Hey, I am here.
And there you are. All right, great. I just couldn't find you.
Hi, thanks. Thanks for a wonderful presentation. I think the paper is absolutely fascinating, and as you mentioned, pretty application driven, but also uncovers lots of territory for interesting fundamental questions to be followed up on. The thing that I keep asking myself is, when we use a therapeutic, we like it to be free of uncharacterized impurities. In fact, we expect that. And what I'm wondering is, what is the full proteome of the vesicles that are produced, sorry for the dog barking, produced by the CNP process? What other transcripts might have entered those vesicles from the cells? And this dovetails a little bit with the question that Tom had, but I'll let him ask that question. But that's what I was wondering.
Yeah, no, it's an excellent question. I think it was actually brought up. There was a News and Views piece done on this paper, and they raised that issue as well. I think it's an excellent question, but it's a testable question, I would imagine, because you can look at how the EVs from different cell types, the RNA composition of those EVs. And so, yeah, if you were going to use this, there would be that quality control mechanism, if that's what you're getting at. Would the response change from one cell type to the other based on something else that was upregulated in various cells versus others?
Does that answer your question, Brian? Do you have any follow-ups?
He says great answer at the bottom, so.
All right, well, we'll move then to, go ahead.
It was a great answer,
go ahead.
thank you. No, I think, I was just thinking, you might have a certain number of genes that end up, I think there's reason to believe that mRNA molecules that are not intentionally introduced may end up in these vesicles as well, and that those could also be translated to proteins, and if the function of those would be very important to know, I think, before it becomes a therapeutic. But I think it's fascinating.
Your assumption, though, is that these stray mRNAs and their proteins are coming from the cell source itself, from one cell.
I'm wondering, until proven otherwise, I would say I think there's evidence that EVs can contain mRNA molecules, and that in some cases those may be able to be translated. So that's what's driving my question. But you're right, that's what I'm wondering.
Yeah.
So we had a question from Tom as well, and there was a little bit of discussion around that already in the chat box. Tom, please ask your question about the RNA sensors.
Hi, nice talk. I was wondering, if you electroporate such massive amounts of RNA into cells, does that trigger any cytosolic RNA sensors? And could that lead to activation of the cells and cytokine release? And related to that, could that also change the loading of endogenous components into these EVs.
Mm-hmm. So there was there was an earlier paper that I referred to when he was looking at the 3 transcription factors being simultaneously or sequentially transduced, where I think he talked about that issue independent of EVs. In this paper, he really doesn't go into looking at dose dependency of the plasmids themselves. The only thing I can say about immunogenicity is that he did see in one of the graphs, there was a, I think it was IL-6 he was looking at. IL-6, no, that was in the, yeah, that was not in the electroporated cells, that was in the uptake of the vesicles. So I don't think he's looked at that in this paper. I think he did discuss that, although I'm not sure again how thoroughly he looked at it in this earlier paper to the 3 transcription factors that he originally presented in this work too. Sorry, I can't give you a better answer than that. It's a good question.
Yeah, I really wonder if that would change the effect if you use these EVs therapeutically. Like, could that have any negative side effects?
Oh, the EVs themselves causing reactions. Yeah.
Yeah, potentially. You don't know, but
Yeah.
that's work for the future, I guess.
Yeah, that's a very good point. You know, whether the function of these EVs is actually changed under these circumstances, and I think that a related question now is coming from Lilia. Lilia, do you want to ask about your viability question?
Yes, thank you for this very interesting and detailed talk. Actually, I was just wondering about how to control the system in order to know whether cells proliferate or they're dying, and what kind of the mechanisms we still got together behind this EV production. But also there is another thing that came to my mind when I actually made this question. Was the system at some point simplified somewhere in the beginning? For instance, just like imagine that, and this is like, if I understand it right, it's electrochemical cell where you can basically put the matrix, the agarose matrix, for instance, with the cells, they grow, and then you apply the different voltage on them, right? And then so and I think that if the simplified system was actually made before, that could actually answer my question, that we could know how many cells exactly were seeded and how do they proliferate, and could we actually control by any staining with annexin fine or anything else what's going on, do they differentiate? Because I've seen 84 hours, which is not that long. Than normally for some of the systems people apply even longer time for the EV production, and that is like more kind of physiological in some of the cases. I don't know if I could talk about it. So yeah, sorry, this is like a question with the answers, but could you maybe comment it somehow?
You raise a very good point, and so let me come back to the cell surface itself. When Jim Lee was first doing these experiments where he was using optical tweezers to locate a cell in a microchannel up against a nanochannel, he was doing that. It sounds like a very sophisticated method, but he was doing it because he wanted to get good contact between the cell and the nanochannel. If you think of this as a circuit, if that cell isn't properly placed, there's going to be a short in the circuit itself, and the cell is not going to electroporate. In his more modern manifestation of this instrument, I think he's just putting the cells on the silica surface itself. I looked in this paper, and I don't know if anybody else found it, but I didn't see any coating or anything else other than the cells are adhesive and they just stick to the surface. that also is a limitation of the system. And so some of these points that you mentioned of viability of cells, different cells on these surfaces, and can the surfaces be modified is an excellent one. It's not one that that was addressed in this paper for sure.
Okay, thank you.
Great, thanks for the question. Wyatt, I think Wyatt had a question that arose from those comments, and then I'd also like to ask for Wyatt's comments on the NTA and DLS calibration.
Okay, so Mike enjoyed that a lot. Do you know why they went through the time and trouble to set up an optical tweezer set instead of just using a gentle pressure force to hold the cell?
Okay, the cells?
Yeah, because if I was doing it, I just put a beaker of water a couple inches higher than the chip and do it with gravity.
Well, first of all, to make sure that only one cell per chamber gets in and that the fit is tight. But then the other more practical reasons is another person in our center was in the center at Ohio State, was a physicist who was an expert at moving things around with optical tweezers.
Okay, so you had a hammer, so you found a nail.
You used it, yeah.
All right. And then, so there was a discussion about particle counting and using DLS to do particle counts.
Yeah, I was surprised when I heard that.
And so basically the way that works is you have to calibrate your DLS with a certain size particle. And then, so you measure the intensity of the gross scatter, and then you use that as a measure of the particle and particle count by calibration. But in order to do that calibration appropriately, you have to have a particle that has the same architecture. Right. And so if you're calibrating with a polystyrene bead where your refractive index mismatch is gigantic and you have a solid particle, you're going to get a ton more scatter per particle at a given size. Whereas in EV, the refractive index mismatch is to first order just the membrane. There's a slight mismatch in the interior compartment, but it's largely aqueous. So unless you make a serious correction in that calibration, your DLS particle measurements are off. And then now if we add into it the complications of heterogeneity in size, where different particles of different sizes produce different amounts of scatter, it just gets to be a very hairy problem that's hard to untangle.
Yeah, and the other issue with that too, Wyatt, is so NTA is being used as the standard with which to calibrate the DLS, and he's showing particles that are 35 nanometers in diameter. He's getting down to where the NTA is starting to really lose resolution.
So I mean, a number is better than no number, but just understand that number is a very fuzzy number. In our hands, different particle counting techniques, we're lucky if we get them to match within an order of magnitude.
Yeah.
And that's something that we're working on very actively to try to harmonize amongst.
But the relative effect should be better than on an order of magnitude.
Yeah. If you're just doing a ratiometric measurement, then that's much more fair.
Right.
But the absolute quantitation is still something that we don't know how to do. We're actively working on how to do that across NTA, cryo, DLS, AF4 mols, and spectrodyne, and flow cytometers.
Yeah, I agree completely. I think to say that 1 million EVs are being produced per cell and 75,000 is being produced per cell by starvation, that's a stretch, but saying there's at least an order of magnitude, maybe 2 orders of magnitude difference in the EVs being produced, that's believable to me.
I agree with you.
Excellent. Well, thanks for those comments, Wyatt, and thanks for the discussion, everybody. I'd like to thank you all for joining today, and I'd like to thank Mike for sharing his expertise and his insights with us. I do want to note that there will be a webinar tomorrow of the Extracellular RNA Communication Consortium of the US NIH. So this is, I believe, open to everybody. You can go onto their website, which is xrna.org, and look up the details of this. We're going to have a talk from Sheng Zhang of University of California, San Diego, who's going to be talking about sequencing extracellular RNA from very small amounts of biofluid. So I think that'll be an interesting seminar. So you can look that up on xrna.org. And with that, I'll say goodbye to everyone. Thank you again. And we look forward to seeing you next week. Take care, all.
Thanks, everybody.
All right.